Okay, Team Bio, I realize these notes are getting a little crazy, and I apologize. Maybe I should make them less hectic. It's a trade-off between trying to put all of the information that we've studied out here for you so you can see the connections, and also simplicity. So I've chosen hectic over simple, but hopefully this is going to you're going to see how it all connects together. So obviously we've been studying cellular respiration. Both aerobic and anaerobic start the same way. They start with glycolysis, which produces a tiny little bit of ATP and occurs in the cytoplasm. If there's no oxygen, which we've talked about in great detail because our yeast fermentation lab was anaerobic, without oxygen. Our yeasts were doing alcoholic fermentation. If you were without oxygen, you would do lactic acid fermentation. You could only do this for so long before you expired, however. But anytime you exercise and you go anaerobic, you work out hard, you produce lactic acid because you are doing cellular respiration in the absence of enough oxygen. Okay. Today's screencast is going to focus on the aerobic pathway. What cells do, which what they prefer to do, is in the presence of oxygen, break down the products of glycolysis. And we're going to cover the Krebs steps and the Krebs cycle. This is going to produce a little bit of ATP for us, as you will see. And it involves the serial breakdown of glucose. Okay, so glycolysis, this was 2 nights ago. Fermentation is what we did in class today. You know this is the anaerobic pathway. And instead, we're going to go down the aerobic pathway because we have oxygen. Okay, we're going to move from glycolysis, which takes place in the cytoplasm, now to the mitochondria, the matrix of the mitochondria, which is the very interior, I don't know the picture of the mitochondria, the very inner part of the mitochondria, to what we call the Krebs steps. And here we have pyruvate, which was the product of glycolysis, enters the Krebs steps. So pyruvate, one pyruvate molecule joins with something called coenzyme A and NAD plus to form acetyl-CoA, one molecule of CO2, and one NADH. But bear in mind, glycolysis involves breaking up one 6-carbon molecule, glucose, and creating 2 pyruvate molecules. So the prep steps happen twice. So for every molecule of glucose, 2 pyruvates are created, and every pyruvate goes through the prep steps. So the prep steps happen twice per molecule of glucose. So pyruvate joins with coenzyme A and NAD plus to make this molecule called acetyl-CoA, one molecule of CO2, and a molecule of NADH. Now, this CO2, this is what you exhale. So CO2 produced in all of your cells is gathered in your bloodstream, it's brought to your lungs, and then when you breathe out, you're breathing out the CO2 that in part you've created in the prep steps. Okay. The NADH is going to be saved for later. We put a blue box around it because it's going to be involved in electron transport phosphorylation, the last part of our system. Okay, acetyl-CoA enters what we call the Krebs cycle. It's also sometimes called the citric acid cycle. And the Krebs cycle, or the citric acid cycle, has to spin 2 times for I guess to combust or break down 1 molecule of glucose. Okay, and the Krebs cycle is all about redox reactions. And you remember our acronym, LEO goes GR, loss of electrons is oxidation, gain of electrons is reduction. We are going to be stripping electrons away from this high energy molecule that, remember, this originally was glucose and it contains high energy electrons on it. So we're going to take those away and use them to create ATP ultimately. Okay, so oxaloacetate joins with acetyl-CoA, this is a 4-carbon molecule, joins with a 2-carbon molecule to create a 6-carbon molecule called citrate or citric acid, which is where the name citric acid cycle comes from. This 6-carbon molecule is broken down after a redox reaction happens. So NAD plus swoops in here, it picks up an electron from citrate and it becomes NADH. Yes, in that process, a molecule of carbon breaks off of citrate or citric acid, and this is CO2. Just like the CO2 molecule that we created in the prep steps, this CO2 molecule will eventually be exhaled. So on your next breath out, you can think, maybe that molecule of CO2 was created as I changed citrate into alpha-ketoglutarate. Okay, so we stripped a high energy electron away from citrate. We created a molecule of CO2, and now we became a new 5-carbon compound called alpha-ketoglutarate. Okay, the same thing occurs again. We have another redox reaction where we strip a high energy electron away from alpha-ketoglutarate, and we produce another molecule of CO2, which will eventually be exhaled. So on your next exhale, you can think, maybe that molecule of CO2 was created as I changed alpha-ketoglutarate into succinyl-CoA. In between this next step, succinyl-CoA and succinate, we have a molecule of ADP and phosphate swoop in here and use the energy of this compound to form ATP. Succinate turns into another molecule called fumarate with another redox reaction where FAD becomes FADH2 by picking up electrons, high energy electrons. And then to turn fumarate into malate, we actually have to add a little water. And then when we go from malate back to oxaloacetate, we do another redox reaction where we have NAD plus swooping in here to pick up more high energy electrons, and it's converted to NADH. And then we're back to the start of the cycle, where another acetyl-CoA can join oxaloacetate to make citrate. So remember, this has to spin 2 times per 1 molecule of glucose because 2 pyruvate are created from 1 molecule of glucose. So if we count up all the CO2 molecules that we produce, the prep steps happen twice, so that's 1, 2 molecules of CO2, and then 3, 4 molecules are created in 1 round, and then 5, 6 are created in another round. So you can see here, we took this 6-carbon compound and we turned it into 6 molecules of CO2. The CO2 is a waste product, but what we really care about in this whole process is NADH. These high energy electron carriers are dropping off high energy electrons at the next step in the cycle called electron transport phosphorylation. And so you can have all of your high energy electron carriers here coming in. Oops, I did that the wrong way. They're dropping off electrons. And this is where we're going to make most of our ATP energy later on down the line. Okay, so let's do some overall accounting. We know that in glycolysis, on net, we make 2 molecules of ATP and 2 molecules of NADH. In the prep steps, we make 2 NADH, and in the Krebs cycle, because it spins twice, we make 2 molecules of ATP, 6 NADH, and 2 FADH2. So overall, I told you that glycolysis produced 2 ATP, and all of this, the aerobic cellular respiration, produces on net 34 to 36 ATP molecules. So you're going to see next screencast how these ATP molecules are actually synthesized. The most ATP of this whole system is created in our last step, electron transport phosphorylation. Okay, that's all.